Hello everyone, I am Dr. K. Sudarshan, working as an academic counselor at Indira Gandhi National Open University, and I've been there for the past 3 years. I've been taking the theory and the practical classes and also being a project guide for food microbiology. In today's video, it is a short video on food microbiology. Let me get into the session. Microbiology. We all know what is microbiology because of this pandemic situation. It's the infection of the virus and the disease spread. So microbiology is the study of small microscopic organisms which are unable to see through naked eye. You require a microscope to look their morphology and the cell organelles. There are many advanced techniques of microscopes. We can get a clear picture, such like scanning electron microscope and transmission electron microscope. Coming into the microbiology, they are present everywhere. They can be present in the volcanic eruption in the rock, the hot springs, the thermal vents, or also in the glaciers of Himalayas. We categorize them based on their habitat, which we call as a halophiles, bacteria which likes to grow in the salt condition. It may be from the minimal to maximum level. They can adapt and they can multiply. Acidophils. They can survive in many acidic medium. They can infect the fruits which are acidic too. Alkalophilic. They like to live in the base conditions. And barophils, they can adapt to extreme pressure. And there are several disciplines in microbiology. A study of bacteria, which we call as bacteriology. The study of fungi, we call as mycology. A study of algae, we call phycology. And the study of virus, we call virology. And there are many specializations in microbiology, which are medical microbiology. We study about the immunology and the medical importance of the microorganisms, and agriculture microbiology, which will give you a very good idea about the crop protection and management from the disease spread. Industrial microbiology. It's about the scaling up of microorganism where we can get the desired product out. Then about the probiotics, which you all know about the importance of the gut bacteria in our intestine. Suppose if we are eating green leaves or the vegetables, we don't have the enzyme to digest the food. Example, if you take cellulose, we should have cellulase. But if we lack that microorganisms, example, you can take lactobacillus or E. coli or bacillus. E. coli are supposed to synthesize the cellulase enzyme which will digest our food. Okay. So thereby we can inculcate this microorganisms by taking the probiotics. Coming into food microbiology, it is the study of microorganisms in food spoilage, foodborne diseases, food production, food preservation. The main quality of a food microbiologist, you need to have a clear understanding on the cell structure, morphology as well as anatomy, and he needs to have a control on the growth factors. What are the factors which affects the growth, and about their benefits and the demerits. She called the merits and demerits of microbiology. Till 1500, we don't have a clear idea about microbiology, and many scientists, example, you can take the cell theory, who formulated cell theory, Theodore Schwann. He stated as a microscopic plant that is causing some changes that will bioconvert into alcohol. Later, the father of food microbiology or the modern microbiology, we called Louis Pasteur. He coined the term microorganisms, which are responsible for the alcoholic fermentation. And later, many scientists involved, and it all came into limelight in 1900. Development of many instrumentation, development of many advanced techniques to isolate, to culture. The three main categories of microorganisms. They are very small and their size ranges from 300 nanometers or even very lesser. And recent advancement, they're also bigger in size. Example is Thiomargareta species report to have larger in size of 2 centimeters. Okay, but comparatively, the cell structure, the arrangement, and the specialization of the cells are very less compared to a plant or animal. And third, you can culture them or isolate them very easy using simple laboratory techniques. Then you can also classify a bacteria into prokaryotes and eukaryotes. Prokaryotes, the word is primitive, karyon means nucleus. You don't have a definite nucleus. Okay. And eukaryotes, eu means true, karyon means nucleus. Yes, you have a definite nucleus for that. The membrane organelles are present. It's a basic one. And next, about fungi. You all know the cell wall of fungi is made up of chitin. It is a biopolymer. So repeated structure of many monosaccharides or monomers we call as a biopolymers. And fungi, normally we use to stain a microorganisms and do example gram staining for a bacterial cell wall. And for the fungal, we use a special stain which is called as lactophenol cotton blue. And in virus, yes, they are non-living as well as living. They are obligate intracellular parasite. It requires an host to multiply and to live. Yeah. It's about the parasites or the symbiotic association. Example, lichen is one association with fungi and algae, which are called as a pollution indicator. Coming into food microbiology. As we said, it's a study about the foodborne diseases. Yes. Water is acting as a vector for the disease spread. Example, if your food is being produced with an infected water, yes, it can pass the enteric diseases, things like typhoid or cholera, amoebiasis, helminthiasis. So we need to have a very clear understanding on the infection and what is the role, how to identify the microorganisms using. Nowadays we also have very good techniques, very instant technique like DNA sticks is also available. Many nucleic acid probes are also available. Within few minutes to seconds we can identify the causage of the disease spread. Next, coming into food bioprocessing. Bioprocessing is the word related to fermentation. When the food gets fermented, so nowadays people likes to take fermented foods through microorganisms. It can change the substrate into a value-added products. And biopreservation, called the food biopreservation. If you know to control their growth study, that is the growth cycle, you can control the shelf life. A biopreservant is the one which will increase the shelf life. And people nowadays having great interest on consuming less preserved substances. The usage of less preservative or without preservation. Food additives. If you take food additives, yes, there are many advancements in food additives. Example is color and the stabilizers. The ice cream which we are eating has been added a special polysaccharide material which is called as carrageenan, which is also a byproduct from microorganisms. And probiotics, as we already said, probiotics, how important for the maintenance of our gut, the digestion in our stomach. So So this is about a short video on food microbiology. So I welcome you all for the course and get benefited out from this regional center. And I'm Dr. K. Sudarshan, working as an academic counselor in Regional Center for Chennai National Agro Foundation. Thank you all for joining with me, and thank you for giving me the opportunity to introduce about myself and about the subject. I hope it will be helpful. Thank you.